Hello everyone. In this session we are going to discuss about the chapter phylum Porifera under the paper Animal Diversity 1, that is non-chordates. So in the previous classes we have discussed about the general characteristics of sponges and their classifications. So in this session mainly we will concentrate on histology of the spicules and canal system in sponges. So what is these spicules now? So spicules are nothing but the skeleton of the sponges. So according to Haeckel, he was the person who distinguished the skeleton of sponges on their majority of their skeletal basis. For example, spicules, spongin fibers, or combination of both he classified. So structure and type. So spicules, they are mainly microscopic crystalline structure and they also are very much rigid and their form. And these spicules consist of spines and rays, and usually they are secreted by special mesenchymal cells called as scleroblast cells. So in the last session I have explained about the three layers: outer ectoderm, inner endoderm, and middle mesenchymal layer or the mesodermal layer. So these spicules or the skeletal rigid structures, they are arising or secreted by mesenchymal cells. So on the basis of the deposition, there are different types. One is core of organic material or the calcium carbonate or colloidal silica. So first, calcareous spicules. So these spicules, they are mainly made up of calcium carbonate or silicate. So these are belonging to the class Calcarea. So what are the three different types of classes? Class Calcarea, class Hexactinellida, class Demospongia. So in that, class Calcarea belongs to the calcareous spicules. Next, siliceous spicules. So in the picture you can see the Euplectella, they are glass sponges. So they are made up of the silicon, and these spicules are having the characteristic of Hexactinellida. So these are the two different types depending on the deposition on core organic matter. So next, on the basis of their size and function. So on the basis of their size, we are having two different types, that is megasclerates, microsclerates. So megasclerates are much larger in size and the skeleton will be of sponge body. But whereas micro, they are very much small. So now if you can see in this picture, the microsclerites are very much smaller when compared to the megasclerites. So these are the microsclerites. So these all are the megasclerites. So megasclerites, they are much larger, microsclerites are smaller. So on the basis of number of axes and rays, they are classified into different types. So first one, that is monoaxon. So monoaxon means the spicule at one point it will be pointed and at one point it will be blunt. So it is monoaxon types. So these spicules are mainly formed by growth along at one axis, and they may be straight, needle-like or rod-like, may be curved also. So at the end, as I said, it will be pointed at one end, hooked or knobbed, and mainly they both are calcareous and siliceous type. So under that you will be having monoactinal, diactinal. So monoactinal means what happens? It only grows in one direction. Diactinal, it grows in both the direction. So let's see some of the pictures while coming. So next, tetraxon. Tetra means what? 4 rays. So usually tetra means 4. So these spicules are having 4 rays, each pointing in different direction, and one of the 4 rays is elongated, which gives a crown of 3 rays. So they are also called as trianes. And later you are having all the 4 rays persist. They are also called as tetractinellidae, or it is also called as the quadrate type. So tetraxon, it is also called as tetraradiate or quadraradiate. So if these are elongated, and it is also called as amphidisc type. Next you are having triaxon. Tri means 3. So usually triaxon means 3 pointed axons, they are merged together. So minimum 2 has to be there, and they belongs to hexactinellida. So hexactinellida means what? 6 belongs to, or it consists of 6 rays. So minimum 2 triaxon has to be kept side by side, or not side by side, one upon the another, then it will become 6 rays. Polyaxon, very several equal radiation from the central point, like maybe you can make out like from one point all sharp needles are diverging, so that will be polyaxon. So this is the picture. You can see monoaxon, it is a single type, but it is also called as diactin, means both this side it is growing. And triaxon, it is having 3, and hexa you can find 6, then tetra 4, polyaxon you can find many. So these are the different types of the spicules which are the making rigid structure of these sponges. Next, very important, that is canal system in sponges. So as you can see the picture, these are the 3 important types of canals, that is asconoid, syconoid and leuconoid. So very important for your examination point of view. Any 2 type can be asked for 7 marks, but all the 3 types has to be studied. So any 2 type they can ask. So what are these canal systems then? So canal systems are nothing but they are the cavities of the body which are transferred by the currents of water, and they nourish the sponges from the time it enters the pores and it passes out through the osculum, and which is collectively called as the canal system. So as I said in the previous classes also, the ostia is nothing but the small pores which helps in the movement of water from one place to the another place inside the sponges, and later on it mainly the upper part, that is osculum, the water is thrown away. So this high degree of complexity which is seen in the sponges, so they are the mainly primitive type folding of walls and accompanied by restrictions of conacocytes or collar cells. So that is about the canal system. Let's see one by one what are the different types. So as I said, the types are asconoid, syconoid and leuconoid. So first one, asconoid type. So as you can see in this picture, asconoid type of canal system is simplest of all, and it is radially symmetrical. So if you cut it into half, so it is radially symmetrical, and they are having the large central cavity called as spongocoel, and at opening larger part that is called as osculum, and the walls are mainly composed by outer and inner epithelium, and in between you will be having mesenchymal layer. So this outer epidermal layer, it is having the single layer of the flat cells, and the inner epithelial layer it will be having the spongocoels. The mesenchymal layer consists of skeletal spicules. Several types of amebiocytes are also mainly present, and that will also help, or it looks like the asconoid sponge for perforated, and later incurrent pores of osteoids are also present, and that leads to the pores of intracellular, and this canal through tubular cells, they are also called as porocytes. So porocytes are nothing but the small hollow cells which mainly helps in exchange. So this is how the water enters from the exterior to the inner pores, and spongocoels, osculum, it is thrown out by the body. So here also food will also pass, and metabolic wastes are also thrown out. So this is about the first type, that is asconoid type. Next we are having syconoid. So cyconoid is nothing but small modification in the asconoid. Here overfoldings are there and finger like projections will be there, and they are radial canals. They are called as radial canal because it stores the water for some time, radial canal systems. So as you can see in the picture, the radial canals are the free projections, and the outer water surroundings, they mainly helps in the movement of water. And this wall of radial canal, they fuse in such a manner that what happens, the tubular space is present, and due to this tubular space, the incurrent canals and the open exterior up and the blind outer end of the radial canals by the aperture. So that is termed as the dermal pores. So dermal pores are mainly present in each radial canals, and there the exchange of water takes place. So how the water is thrown out? So water is thrown out in such a way that it enters to the dermal pores, incurrent canals, and radial canals, internal ostia, spongiocele, and osculum, and it is thrown out. So this is how water transmits from outer region to the inner region, and later it is thrown out. So there are some of the difference. So make a note on that. They can be asked it for 3 marks. So if you know about cyconoid and asconoid, you can write the difference between them. So next, the last one, that is leuconoid type. So leuconoid are the major modification of both asconoid and cyconoid. So here the conacocyte layer or radial canal of cyconoid stage, they invaginate into many small chambers. So you can see there are small chambers in inside the canal system. So these are nothing but the small canals everywhere. So inside you can find the small canals, and these small canals, they are very much rounded and oval flagellated chamber, and later these conacocytes are mainly limited and mesenchymal fills with the space, and later that spongiocele will be very important and it holds the water, and whole sponge becomes irregular in structure. And due to that, the interior of sponges become permeated by many incurrents and excurrent canals, and that leads to formation of space, which leads to the oscula. And the surface is covered by epidermal epithelium, and it has got many pores called as dermal pores or ostia and oscula. And the main characteristic of leuconoid canal is limitation of conacocytes into small chamber. And here, as I said, you are having the apophysis, that is flagellated chamber, which opens into aperture, that is called as apophyles. They are mainly helping in the movement of water. So this is how water transmits from one region to another region. So ostia in current canals are mainly present, and how the current of water flows from the prosodus to prosopylus, and flagellated chambers, apophyles, aphodus, excurrent, incurrent, large canals, osculum, and also that is throwing out. So this is how the water transmits from one region to another region. So they are also irregular in structure because the small chambers which are growing inside, they make space in between them, and the size of the structure of this canal system, it will be irregular in shape, and that leads to the irregular structure, and flow of water is fairly rapid. So due to that, 3 stages are there: euripylus, aphodal, and dipodal. So not necessary to study, but I am going to tell about one more type, that is Ragan type. So Ragan type is nothing but the 4th type of canal system, but this is not there in your syllabus, just I am making a note on that. So it is a calcareous sponge which is a part of the last one, leuconoid. So here the demospongia is derived from the stem stage termed as Ragan, which raises the direct rearrangement of the inner mast cells. So here what happens, the Ragan type, the sponge has the broad base. As you can see, the base is very much larger, called as hypophyre, and hypophyre, that basal wall is called as hypophyre, which divide the flagellated chambers, and spongiocele is broader and it is somewhat oval flagellated chamber, because you can see it looks like a funnel type, the spongioceles, and thickened mesenchymal cells are mainly present, that leads to the formation or leads to how the water can transmit from one region to another region. So these are the different types of water canal systems and the spicules. So in this mainly chapter of the Porifera, we have started with the general characteristics of poriferans, the characteristics, and later we have studied the characteristics of their classes and spicules and the water current system or water canal system. So all these are important for your examination point of view. So that's all about this session. Thank you.